So I'm going to be talking about something a little bit different to Olivier, but it's nonetheless, I think some of the background that Olivier has covered is definitely relevant, and I hope to show you in some different ways some of the same concepts early on, especially for the family members in the audience. So what I'm going to be talking about today is some work that we published a couple of years ago, but we have started to move on and follow up some of those findings. And I'll talk in the last couple of slides about our current efforts that we've been sort of taking with some recent funding that we've acquired from GRIN, in fact. So most of the work I'm going to talk about today was funded by the Medical Research Council. I'm just finding the laser pointer, clicked the wrong button, sorry. It was funded by the Medical Research Council in the UK and was performed at the University of Sussex. So to give you an overview of the sort of three areas of things that I'm going to cover today, I'm going to cover a bit of background on GRIN variants, NMDA receptors, and their role at synapses. I'm going to talk about how GRIN2B variants affect synaptic NMDA receptors, in particular some of the work that we've been doing to establish our understanding. And I'm also going to talk about some of the potential implications of that work and the sort of directions that we're moving in at the moment. So GRIN genes are part of a larger family of related glutamate receptor genes, which the community in this room and beyond have called GRIN. So we have the GRIN subunits, or the NMDA receptor subunits. We also have related genes, the GRIA genes, the GRIC genes, and the GRID genes. So these are the AMPA, the kainate, and the orphanal delta receptors. So today my talk is going to focus on GRIN genes, in particular the GRIN2B subunit gene. So in this audience, the families are here because they have a child who has a genetic variant in one of their GRIN genes, and what that means is that there's a change in the DNA, and it's the part of the DNA that encodes one of those GRIN genes. So we've got, in particular, I'm going to show an example, GRIN2B. So when there's a change in the sequence of the DNA, in this case of the GRIN2B gene, what that can do is that sequence change is also represented in a temporary copy of the gene, which is formed and used to code for protein. And the protein in this case is actually the GRIN2B subunit. So this is an example of what Olivier had on his slide, a missense mutation or missense variant. So it's actually changing the type of amino acid in the protein. So it's changing this little part of the protein, and this can affect the structure and ultimately also the function of the receptor in some way. So we all have 2 copies of the GRIN2B gene, and individuals with GRIN2B disorder actually tend to have a disease-causing variant in only one of those copies of the gene. So in fact, we'd have 2 versions of this, and you'd actually have maybe this version, which is the sort of wild type or normal, and then this is the variant form. And so you end up getting 2 copies of the protein that are different. Parents of children with GRIN variants don't usually have a GRIN variant themselves, so we refer to it as being de novo. It's not been inherited from the parent, although that is possible, it's just rarer. And variations in the GRIN2B sequence usually occur in the germline, so the sperm or the egg, so that all the cells in the affected child have a copy of that GRIN2B variant. That's typically what is found. So it's not that GRIN variants are just suddenly appearing. It's more that we're discovering them thanks to sort of advances in technology. So it was only sort of around the year 2000 that the human genome was actually sequenced. And it wasn't until around 2015 or so that whole exome sequencing, which is the exome is the protein coding part. So if you think of the genes that encode proteins like the GRIN2B gene, it only then did it become sort of economical to actually sequence the whole exome in an individual to help sort of identify genetic changes that might be leading to rare disorders. And so over those years, of course, things get cheaper and cheaper, and especially the more widely they're adopted, the demand on it is an incentive to make these things more economical. And so the number of variants that have been discovered year on year has accumulated and is, I believe, is still rising. And some work by, I think, Johannes has actually predicted that the incidence of disease-linked variants in GRIN genes is actually much higher than we would have expected. And so this is of any particular GRIN gene, so all of the GRIN genes together is expect a sort of an incidence of about 1 in every 5,000 births, which is actually much larger than any of us would have imagined. So the effect that these sort of missense variants that change amino acid sequences in the GRIN genes or in their proteins, the effect that they can have on the proteins can vary. Olivier talked about how the genetic variant might not cause a change in the function of the protein itself, but it could actually affect other aspects of the protein's sort of trafficking, and that ultimately affects the function in a biological context, but not necessarily the direct function of the protein. And I'm going to be talking about variants that do affect sort of the function of the protein more directly. So we think of, we know NMDA receptors are in fact ion channels. So ion channels, they sit in the surface, on the surface of neurons, and they allow the passage of, in this case, for the NMDA receptor, positively charged ions, which carry electrical current across the membrane. And normally these ion channels have a receptor on them that for the glutamate neurotransmitter. And essentially this receptor is normally, the channel is normally closed when the neurotransmitter is not bound, but it can open in response to neurotransmitter binding and allow current to flow. And so current flow is illustrated here in this little insert, and that's sort of, I'll be showing how sort of the receptor activity changes over time using these sorts of graphs as we go through the talk. So normally the receptor can open and close on demand, so it's almost like a traffic light for the flow of electrical current. And if the variant is causing what people call a loss of function, it is inhibiting essentially it can be inhibiting the flow of current through the channel in different ways. It might make the channel stay open for less time, or it might make the current smaller overall, the amount of current that can pass through. And you can get gain of function variants where essentially too much current is flowing through the channel, and potentially some of the current is actually represented as calcium ions, and that's very important for cell signaling, and Olivier talked about that and the consequences that that can have on downstream functions that the receptor is known to have. And so essentially these currents can be bigger, and the channel can stay open for too long. So this is the sort of classification when people are talking about gain and loss of function variants. When you're getting a functional classification, this is the sort of researchers are looking at properties of the receptor that ultimately lead to changes like this. So it's a little more complicated than that for the receptor. So I talked about for the NMDA receptor, I talked about neurotransmitters. So the glutamate neurotransmitter binds, and that's what it gates the opening and closing of the ion channel. There are actually other factors involved to ensure that the channel can open or to prevent it from opening. So we have the coagonist binding site, so glycine or D-serine. Many of you would have heard of L-serine, so L-serine is the precursor for this coagonist D-serine, and binding of that is also required for for the opening of the channel in combination with the glutamate neurotransmitter, although this site is usually occupied with the coagonist. There's also a magnesium ion that's blocking the pore of the ion channel, so preventing current flow, and it's necessary for that to come out as well for current to flow through the channel. And usually sort of neuronal activity, or what we call depolarization of the membrane, is what is responsible for expelling that magnesium ion. So when there is a genetic variant and it's causing either gain or loss of function, it can be affecting multiple things. And this is just some examples. So it can be affecting either the binding of the neurotransmitter glutamate or the glycine or D-serine. It could be affecting the unbinding of the magnesium, making it either easier or harder for it to unbind during neuronal activity, membrane depolarization. Or the variant can actually directly affect the coupling between and the opening and closing of the ion channel, even when all of these factors are happening normally. Or it can be a combination of all of those things. And it could even be a combination of those things and the fact that maybe the receptor doesn't even make it to the surface, and so effectively like a null variant or knockout of the receptor. So the NMDA receptor genes, so they encode for that NMDA receptor. The NMDA receptor, this is the structure of the NMDA receptor. It's actually a complex of more than one copy of the protein that's encoded by the gene. And these receptors are usually present. They're not exclusively, but they're canonically or traditionally thought of as functioning at what we call excitatory synapses, which use the neurotransmitter glutamate and are responsible for signal transmission between neurons in the brain and are absolutely essential for sort of the way that our brains function day to day. And genetic variants, therefore, affect the protein, affect the synaptic function, and the ability for neurons to communicate and ultimately lead to neurological disorder. So NMDA receptors in nervous system development, they are expressed actually very early on in development. I think when we talk about NMDA receptors in the scientific community, a lot of people have a focus on sort of adult roles of the NMDA receptor. But we've got here the human sort of time course of before birth and after birth, and it's sort of aligned up roughly with that of the mouse, which is the model system that myself and others, many others work in. And we see that the NMDA receptor expression shown here by this sort of increase in this sort of I don't know what color you call that, yellow, increase around sort of especially around birth. And actually, NMDA receptors have a really important role in a lot of things. And sort of traditionally, we talk about its ability to help modify synapses and maybe control excitability in the adult. But even very early on in neurulation, neural migration, refinement of synaptic connections, that NMDA receptors play really important roles in them, and they're very early on in development that is happening. I just want to show one example of this before I start talking more about how we fit into this in my research. And that is to just illustrate how important NMDA receptors in development are. So mice that have a loss of function of both alleles of either GRIN1 or the GRIN2B gene have really severe failure to thrive, to put it nicely. And actually the reason is this. So if we look at this is the sort of a diagram of the head of a mouse, and we've got the mouse has whiskers, and those whiskers are really important for the mouse to be able to sense its environment, and also it's really important for it to be able to for something called a suckling reflex, which allows it to these very neonatal mice to feed. And these barrels, we call them, or the whiskers, have their axons from the neurons in those that are associated with those whiskers project to the brainstem, and it's very organized. It's organized into what we call a barrelet field. So essentially each whisker maps onto one of these sort of circles in the barrelet field. And the problem is actually during development, early on, it looks like this. It's very disorganized, and it's really important for it to form this this organization for it to actually, for the whiskers actually to function properly. And so without the ability to form this refinement, which is associated with knocking out these NMDA receptor subunits, we don't get this barrelet field. We get this, it's stuck in this disorganized kind of, without this refinement. And so this leads to defective licking and mouthing onto the nipple. Actually, the pups suffer from nutrient deficiency and suffer pretty badly from that. So when we think about the development, we don't. I was talking about GRIN1 and GRIN2B. Actually, GRIN2A is also expressed to a large extent in the main sort of brain area in the forebrain, so the cerebral cortex and the hippocampus. And to start with, early on, around birth in mice, GRIN2B is very highly expressed, embryonic stage up until birth. And it isn't until around birth in mice that we actually start to see a great deal of the GLUN2A subunit being expressed. So this is a list of all the sort of GRIN genes in the human, and this is the names of the encoded proteins, and I'm focusing on the GRIN2A and GRIN2B here. So if we have a look during development, actually, and looking at the properties, functional properties of these NMDA receptor subunits, we've got the GLUN2A subunit, which is not expressed until the adult. And so we've just got GLUN2B subunit here, which is shown in green. And one important property of the GLUN2B subunit is that it imposes very sort of slowly deactivating synaptic currents for the NMDA receptor component. And as you go through development and express GLUN2A, this accelerates the deactivation of the receptor. So the synaptic currents, the receptor stays open for less time. And so you have a mixture then of these two types of receptor. And what's quite interesting actually is that you can form not just sort of separate GLUN2A and GLUN2B receptors, but you can form a combination of the two where they have a copy of each of the GLUN2A and GLUN2B. And what was really interesting, some work that Casper Hansen, while he was in Stephen Train Ellis's lab, found was that actually these they're called triheteromeric receptors, have properties that really resemble more that of the GLUN2A only receptor. And so this we found interesting because we thought, well, if there's genetic variants in GLUN2B, would they have more effect on the receptors early in development in the absence of GLUN2A? And then really, how will GLUN2A have an effect on the receptor properties? And so that's the sort of the work that I'm going to show you in the next couple of slides. So we have the genetic variant of GLUN2B in these different sort of receptor combinations and how they would be affected by the presence of GLUN2A. And the way that we have done this is to use we use a mouse model system. We take organotypic hippocampal slices. So this is part of the forebrain that's very important for sort of learning, certain types of learning, such as spatial learning and explicit memory formation. And what we do is we perform our genetic manipulations in vitro to try and model these different genetic variants in this system. And we can compare neurons which haven't been genetically altered with those that have, side by side, and look at how the currents, how the current through the receptor at synapses is changing. And what we did is we want to look at how the effect of GLUN2A or GRIN2A is changing the synaptic currents mediated by NMDA receptors when we have changed the GLUN2 subunit to one of its genetic variants. In this case, we're looking at some putative gain of function variants. I'll talk a little bit more about that in the next couple of slides. So what we do is we knock out or remove both GLUN2A and the GLUN2B subunits, which are widely expressed in these neurons, and we replace the GLUN2B with the either wild type or the normal or the or a genetic variant. And we also do something very similar. So to look at the effect of GLUN2A, we do exactly the same experiment, but we don't remove the endogenous GLUN2A. So we allow the normal sort of levels of GLUN2A that are expressed in these neurons to still be there and effectively combined with our genetic variants of GLUN2B that we introduce. So looking at the effects on synaptic currents mediated by NMDA receptors, when we have removed GLUN2A as well, we actually recapitulate quite well the sort of functional characterization that the folks at Emory have done for these receptors. And that is to say that we see these channels that are, or the neurons expressing these gain of function variants are producing synaptic currents which are prolonged. But these NMDA receptor mediated synaptic currents are more slowly decaying compared to the normal or wild type receptor that we introduce. And we also see that if we also see, oh, I've actually put this the wrong way around, so I'm going to have to do this first. We also see that with some loss of function variants, if we do the same with some loss of function variants, in this case I'm just showing C456Y, what we can see is that compared to not introducing any GLUN2B at all, we get we see almost no benefit to expressing this loss of function variant. So it really is behaving like a loss of function variant compared to, say, introducing a normal variant. So that's all in the context without GLUN2A. So they're behaving like gain of function and loss of function in the absence of GLUN2A. But as soon as we leave GLUN2A in there, these gain of function variants are no longer looking like gain of function variants. So if you compare this to the sort of normal receptor here, the gain of function variants are producing synaptic currents which are actually slightly faster than the normal or wild type. And interestingly, even with this loss of function variant, we are actually starting to get currents more like we're getting in when we replace GLUN2B with a sort of a normal or wild type receptor compared to when we actually don't introduce any GLUN2B at all. So what we're seeing here then is the GLUN2A is making a big difference to the impact that these GLUN2B variants are having on synaptic function. It's also, if we scale the peaks of these NMDA receptors and have a look at the decay, comparing the sort of these gain of function variants with the loss of function variants in the presence of GLUN2A, what we can also say is this is actually the same data, but just looking at it in a slightly different way. So these are the gain of function up here. You can see from sort of the work in the heterologous expression systems that that people have done at Emory, Stephen and Honji, and these are the loss of function variants. We can see actually, despite this drastic differences and the difference we see when we express these receptors without GLUN2A, we can see that actually the effect on synaptic function looks almost identical between the gain and loss of function variants. So this sort of slide is designed to sort of summarize some of those changes and also to highlight actually that when we talk about contexts where we don't have any GLUN2A or where we allow the expression of GLUN2A, we've been looking actually at a stage in development which you might consider sort of later in development. But actually, this condition where you have no GLUN2A expression is actually not too different from what we might expect early on in development. And so this leads us to thinking about, well, could the effect of GRIN2B variants on synaptic function change during development. And so what I'm just going to tell you in the next 2 slides is about some work that I've got a new PhD student working in my lab, funded by GRIN. He started beginning of October, and he's already doing some preparing the constructs and starting to test some of these things out. So it's fantastic development on that front. I'm just going to tell you a bit about how we're approaching this. So if we have a look at that sort of familiar image that I showed you earlier of the changes in the subunit expression during development in the human and the mouse, and this is sort of what a mouse looks like during embryonic development and just postnatally after 2 weeks. The sort of changes in this situation where you've got GLUN2B only and you've got GLUN2A and GLUN2B, really sort of it straddles around sort of the first week of in newborn mice. And so what we're doing is we're trying to take neuronal tissue from sort of late embryonic mice, and we're testing out at the moment a CRISPR strategy that's sort of as an improvement over our original genetic manipulations to knock out the GLUN2B and replace it with a variant of GLUN2B. And then assay it, we then would find the cells which have successfully transfected and undergone this manipulation, and then look at how synaptic function is changing at different stages during development. And so this final slide that I'm going to show you now is really for the researchers in the room. It's not intended for family members to see, but it's more to try and get maybe some feedback from researchers in conversations after this in the meeting. Obviously, I'm keen to be open and sort of take on people's useful experience to try and improve our own efforts. So that, again, this is going to be the work of this PhD student, it's Scott Kinghorn, and he's taking this CRISPR approach that we are basing actually on the sort of knock-in strategy that Richard Huguenot has recently been using in his sort of T-kit system. It's a CRISPR-Cas9 approach, and we're also harnessing the generosity of our folks at Emory, like Honji, who sent through a bunch of plasmids expressed that contain various genetic variants of the NMDA receptor. And we're doing a single-step cloning to generate what we call donor fragments, which allow the knockout and replacement of the first protein coding exon in GRIN2B with a sort of a GFP open reading frame so we can identify the cells, but at the same time allow us to express the GRIN2B variant all in the same all in the same. We know that cells then that are expressing the GFP also have the plasmid expressing the GRIN2B variant. So I'm just going to say a few thanks to various people, including over and above the ones I've already mentioned. So in particular, Marwa El-Mazri, who is a PhD student who produced a lot of the work that I've been showing today. Various people who've contributed in the way, Oliver Steele, Wajiha Aziz, and to Scott Kinghorn, who's starting this new phase of the project funded by GRIN. And my collaborators, Caspar and James, who I didn't show the work of here, but have contributed a great deal to helping us understand a bit, understand what we're seeing using their great techniques. And to Kenji Sakimura, who provided the mice. And obviously, I'd like to thank my colleagues and collaborators at the University of Sussex. And thank you very much to GRIN Europe for inviting me here to speak today. Thank you.